Hello everyone, welcome to my channel Dr. TAPATI'S presentation. Today I am going to talk about nitrogen bases, nucleosides, nucleotides and nucleic acids. This is part 4 of main topic biomolecules. Nitrogen bases. Nitrogen bases are carbon and nitrogen containing aromatic heterocyclic compounds. If you see the structures of nitrogen bases, you will find that these are ring structures and both carbon and nitrogen are present in their ring structures. Therefore, these are heterocyclic compounds. Nitrogen bases are the fundamental components of nucleotides, and nucleotides form the building blocks of DNA and RNA. Later on in this video, I am going to discuss about nucleotides, DNA and RNA in detail. Let's focus on nitrogen bases again. There are 5 main nitrogenous bases classified into 2 groups, purines and pyrimidines. Purines are 2 types, adenine and guanine, and pyrimidines are 3 types, thymine, cytosine and uracil. So total 5 nitrogen bases, A, G, T, C and U. If you see the structure of purine, it is actually two aromatic rings attached together or fused together. So two ring structures fused together in purine, whereas in pyrimidine only one ring structure is present. So name of purine is small, but its structure is big. Whereas in pyrimidine, name is bigger and the structure is smaller. So in this way, you can remember purine name small, structure big, pyrimidine name big, structure small. Another fact is that if you compare thymine and uracil, in case of thymine at the C5 position, methyl group present, and this methyl group is absent in uracil. So there is no only one difference is there in between uracil and thymine. In thymine, extra methyl group present, which is absent in uracil. And in DNA, A, T and G, C, these 4 nitrogen bases are present, whereas if you consider about RNA structure, in that case A, U, G, C, these 4 nitrogen bases are present. So in DNA, instead of uracil, thymine present, whereas in RNA, instead of thymine, uracil present. So this is the difference between nitrogen bases in RNA and DNA. And you have to very easily remember the purine and pyrimidine nitrogen bases. Side by side, you just write down ATGC, which is these 4 nitrogen bases present in DNA, ATGC, and in this way, up and down position, that is A and G, this is actually coming under uh, purine nucleotides, and T and C, these are coming under pyrimidine nucleotides. AT purine pyrimidine and GC purine pyrimidine, but AG purine, TC purine pyrimidine, and obviously uracil is coming under pyrimidine. So in this way, total 5 nitrogen bases are there: adenine, guanine, thymine, cytosine, and uracil. In case of purines, a 6-membered ring and a 5-membered nitrogen containing rings are fused together, and they are numbered in anti-clockwise manner. So in this picture also you can see the numbering starts with this nitrogen, and anti-clockwise the numbering is done 1, 2, 3, 4, 5 and 6, and then again 7th number is there in the 5-membered ring, 5-membered nitrogen containing ring. So this is 7, then 8, then 9, and in this way total 9 elements present in these two rings. And if you consider about pyrimidine, it is a six-membered nitrogen containing ring, and numbering starts with this nitrogen, and it is numbering is like clockwise manner. So this is 1, then 2, then 3, then 4, 5, and 6. So this is actually a clockwise manner numbering, and this type of numbering is an internationally accepted system to represent the structure of nitrogen bases. Nucleosides are made up of nitrogen base and sugar. Sugar means either ribose sugar or deoxyribose sugar. Ribose sugar present in case of RNA, whereas deoxyribose sugar present in DNA molecules. And nitrogen base means either it is a purine or a pyrimidine base. So nitrogen base plus sugar is equal to nucleoside, whereas nucleotides are made up of nitrogen base, sugar, and phosphate. So these three molecules are attached together to make nucleotides. Or in a separate word, you can consider nucleotides nucleotides are equal to nucleosides plus phosphate group, whereas nucleoside is equal to nucleotide minus phosphate group. In my last slide, already I have mentioned that in RNA, ribose sugar is present, whereas in DNA, deoxyribose sugar is present. These are all pentose sugar because total 5 carbons are present in those ribose as well as in deoxyribose sugar. Already I have mentioned that the atoms in the purine ring are numbered as 1 to 9, and that numbering is based on the anti-clockwise manner. Whereas if you consider about pyrimidine numbering, so that is following the clockwise manner, and it is 1 to 6. In order to avoid the confusion of numbering in among purine, pyrimidine, and pentose sugar, the carbons of pentose sugars are represented with an associated prime number for differentiation. And therefore, pentose carbons are 1 prime to 5 prime. Difference between ribose sugar and deoxyribose sugar is that in ribose sugar at C2 prime position OH group present, whereas in case of deoxyribose at C2 prime position hydrogen present. Therefore, it is known as the deoxyribose because oxygen is not present in C2 prime position of the deoxyribose sugar. Only H present instead of OH. At Ignan, let's see nitrogen bases, nucleosides, and nucleotides. Nitrogen bases means those are adenine, guanine, cytosine, thymine, and uracil. These are total 5 numbers of nitrogen bases present. Now, if you consider here, in case of nucleoside means total 5 nucleosides, namely adenosine. Adenosine means adenine plus sugar moiety together is known as the adenosine. So nucleoside of guanine, that is guanine plus sugar, is guanosine. Then cytidine plus sugar, it is cytidine. Thymidine means it is composed of thymine and sugar, and uridine means it is composed of uracil and ribose sugar. And nucleotides are adenylic acid. Adenylic acid means nucleoside of adenylic acid is adenosine, and when adenosine is attached with one molecule, one phosphate group, it is known as the adenylic acid, which is a nucleotide. It is also known as adenosine monophosphate or AMP because only one molecular phosphate group is attached. So it is monophosphate. So if two molecules of phosphate group attached, so then it will be adenosine diphosphate, and if three phosphate groups are attached with adenosine, so in that case it will be ATP. Similarly, guanosine plus one phosphate group is known as the guanylic acid or GMP, that means guanosine monophosphate. Cytidine plus phosphate group is cytidylic acid or cytidine monophosphate or CMP. Thymidine plus phosphate is thymidyllic acid or thymidine monophosphate or TMP, and uridine plus phosphate group, that nucleotide is known as the uridylic acid or uridine monophosphate, that is UMP. For better understanding, I am giving you two examples of nitrogen bases and their structures. Like if you consider here adenine is coming under purine nitrogen base. So two rings are attached together, and uracil structure is only single ring is there. That is six-membered ring. So adenine and uracil, these are nitrogen bases. Adenine present in DNA and RNA both. Uracil present only in RNA. Now nitrogen base plus sugar means nucleoside. So when sugar moiety is attached with nitrogen base, it is known as the nucleoside. And when adenine is attached with ribose sugar, this is not ribose sugar, this is deoxyribose sugar. So when adenine is attached with deoxyribose sugar, it is adenosine. And when uracil is attached with ribose sugar, so that is known as the ribose sugar. So it is known as the uridine. Already I told you uracil is only present in the ribose sugar. So only present in RNA. So uridine means only present in RNA. And a nucleotide in that case, when adenine, then deoxyribose sugar and one molecular phosphate group is attached together. So these three moieties together is known as a adenylic acid. And when uracil is attached with sugar and phosphate group, it is known as the uridylic acid. So these are the different structures of adenine, uracil, adenosine, uridine, adenylic acid, and uridylic acid. Let's know what are the different bonds present in nucleotide. In a nucleotide, pentose sugar is bound with nitrogen base, and pentose sugar is also bound with phosphate group. So total two bonds present in a nucleotide. First bond is in between pentose sugar and nitrogen base, and that bond is known as the beta N glycosidic bond. I have given two examples here, one with purine nucleotide, another one with pyrimidine nucleotide. In case of purine nucleoside or nucleotide, the N9 of a purine ring binds with uh, 1 prime or C1 of ribose sugar or deoxyribose sugar to form glycosidic bond. So this glycosidic bond or beta N glycosidic bond is in between N9 of purine nucleotide and C1 of ribose sugar or deoxyribose sugar. Whereas if you consider about the pyrimidine nucleotide, in that case, beta N glycosidic bond is in between N1 of pyrimidine and uh, 1 prime of ribose or deoxyribose sugar. So that is also beta and glycosidic bond. Second bond which is present in between pentose and phosphate group, it is known as the ester bond. Now basically it is a phosphoester bond because this bond is in between phosphoric acid. So when acid group reacts with alcoholic group, alcohol means in this case C5 carbon of pentose sugar is carrying alcoholic group, and this alcohol acid combination always makes ester bond. I hope already you know that. So in this case also we are getting ester in between phosphate group and pentose sugar. I have given two examples here. One is with purine and another one is with pyrimidine nucleotides. So both the cases you will find phosphoester bond between pentose sugar and phosphate group. So these two bonds are present in nucleotide. Beta N glycosidic bond is present in between nitrogen base and pentose sugar, whereas phosphoester bond is uh, formed in between phosphate group and pentose sugar. This figure will give you a clear picture about nitrogen base, nucleoside and nucleotide. This is adenine nitrogen base. When adenine nitrogen base is attached with pentose sugar, it is known as the adenosine. So adenosine is combination of adenine and pentose sugar. So that is nucleoside. Now, when adenosine is attached with one molecular phosphate group, so together known as the nucleotide, or which is also specifically it is known as AMP, or adenosine 5' monophosphate, or adenylic acid, same thing. But when two molecules of phosphate groups are attached with adenosine, that is known as the ADP, or adenosine 5' diphosphate. And when three molecules of phosphate groups are attached with adenosine, it is known as the ATP, or adenosine 5' triphosphate. Let's focus on nucleic acids. Those are of two types, DNA and RNA. DNA means deoxyribonucleic acid. RNA means ribonucleic acids. DNA is composed of deoxyribonucleotides, whereas RNA is composed of ribonucleotides. The central dogma of molecular biology, or central dogma of life, is a fundamental concept that describes the flow of genetic information within a biological system. It was proposed by Francis Crick in 1958. According to this concept, DNA to DNA formation, that means when DNA molecules are copied to produce two identical DNA molecules, it is known as the replication. And transcription, second process is transcription. So during transcription, a specific segment of DNA is copied into RNA, especially mRNA. And during the translation process, that is the third step here. So translation means this is the process by which mRNA is decoded by a ribosome to produce specific polypeptide or protein. So DNA to DNA formation is replication, DNA to RNA formation is transcription, and RNA to protein formation is translation. So these three processes together we can consider as the central dogma of molecular biology. Let's focus on the structure of DNA and RNA. Nucleic acids are polynucleotides because they are heteropolymer of nucleotides. That means nucleic acids are composed of various nucleotides, and nucleotides are attached through phosphodiester bond. In this figure, you can consider this is the picture of single-stranded DNA. And in single-stranded DNA, there are total three nucleotides, and they are attached through phosphodiester bonds. And 5' end phosphate group is free, and similarly in the at the 3' end OH group is free. So in a single stranded DNA, at the 5' position phosphate group present, and at the 3' position OH group present. But if you consider about the double-stranded DNA, in that case two strands are attached through hydrogen bonds, and two strands are anti-parallel. That means one is from 5' to 3', and similar another strand is complementary to the previous strand. So it is it is anti-parallel, and it is 3' to 5' position. Now, if you consider about RNA, RNA, that means RNA generally single-stranded. So they are also composed of nucleotides, and various nucleotides attached. Two nucleotides actually, two nucleotides attached through phosphodiester bonds. In this figure, total three nucleotides are given. So in between three nucleotides, total two phosphodiester bonds present, and at the 5' position, phosphate group is free, and at 3' position, OH group is free. Nucleic acids are biomacromolecules. Biomacromolecules because it has molecular weight of higher than 800 Dalton. Ester bond formation is occurred in between nucleotide 1 and nucleotide 2. In this case, 3' position OH group present in nucleotide 1 and uh, ester bond is formed with the phosphate group of nucleotide 2. So in that case, condensation reaction is occurred. That means water is removed and due to removal of the water, there will be formation of ester bond. So, in a nucleic acid, a phosphate moiety links the 3' carbon of one sugar of one nucleotide, that means first nucleotide, to the 5' carbon of sugar of the succeeding nucleotide, that means nucleotide 2. And already you know, bond between the phosphate and hydroxyl group of sugar is an ester bond, and therefore as there is one such ester bond on either side, it is called phosphodiester bond. So first bond is here at the 3' position of nucleotide 1, and second ester bond is formed at the 5' position of nucleotide 2. So that phosphodiester bond is known as 3' 5' phosphodiester bond. Already you know about this table, the major complex biomolecules present in living cells. Already I have discussed about carbohydrates and proteins in my previous two videos. In this video already we have discussed about the nucleic acid. So nucleic acids are composed of nucleotides. In case of DNA, it is deoxyribonucleotides, and in case of RNA, those are known as ribonucleotides. And primary bond present between two nucleotides, that is phosphodiester bond, and various elements including carbon, hydrogen, oxygen, nitrogen, phosphorus. These are the major elements present in nucleic acid. Examples: DNA and RNA, that means nucleic acids are of two types, DNA and RNA. DNA function is it stores genetic information, and RNA is required for the protein synthesis. There are more than a dozen forms of DNA secondary structure named after English alphabets A, B, C, D, E, and Z, etc., with unique structural features. That means they have some structural differences among these different forms of the DNA secondary structures. Among them, B form of DNA secondary structure is most popular because it is developed by Watson and Crick. It is described by Watson and Crick, so it is known as the Watson-Crick model. So let's discuss about secondary structure of DNA double helix, or that is known as Watson and Crick double helix model. According to this model, the DNA is a right-handed double helix. It consists of two polydeoxyribonucleotide chains or strands twisted around each other on a common axis. The two strands of polynucleotides are anti-parallel, that means run in the opposite direction. So in this case, you can see one strand is from 5' to 3'. This is 5' to 3', and another strand is from 3' to 5'. So they are anti-parallel. Hydrophilic backbone is formed by the sugar phosphate sugar chain, that means 3' 5' phosphodiester bond. So sugar phosphate backbone you can consider here. This is sugar phosphate backbone. Sugar and phosphate. Sugar and phosphate, this is actually sugar phosphate backbone. So those are hydrophilic in nature, whereas hydrophobic nitrogen bases, these are ATGC, those are nitrogen bases present in DNA, and they are hydrophobic in nature, and they are projected more or less perpendicular to this backbone, but backbone of phosphodiester bond, but face inside due to their hydrophobic characteristics. Now, A and G, G of one strand compulsorily base pairs with T and C respectively. That means A always binds with T and G always binds with C through hydrogen bonds. And there are two hydrogen bonds between A and T, whereas three hydrogen bonds present in between G and C. hydrogen bond formation is only possible between A and T, and in between C and G, that means G and C. And their special arrangement favors two hydrogen bond formation in between A and T, and triple hydrogen bond formation in between G and C. And GC is stronger by about 50% than AT bond, because of the fact that in between G and C triple hydrogen bond is present, whereas in between A and T double hydrogen bond is present. Next characteristic of DNA double helix is that each strand appears like a helical staircase. Left side is a double-stranded DNA structure where helical staircase structure is clearly visible. Each step of ascent is represented by a pair of bases. At each step of ascent, the strand turns 36 degree or 360 degree for a full turn. One full turn of the helical strand would involve 10 steps or 10 base pairs. So in this figure also it is clearly visible total 10 base pair present within a turn. We can count those 10 base pairs. This is 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So total 10 base pair present within a turn, full turn. And the pitch is 34 angstrom or 3.4 nanometers. So distance between this distance of this full turn is 34 angstrom or 3.4 nanometers, and the rise per base pair is 3.4 angstrom or 0.34 nanometers. So distance between two base pair is uh, distance between two base pair is 3.4 angstrom. So all mathematical calculation if you do, it is very clearly visible. 10 base pair distance is 34 angstrom, so distance between two base pair, that is 3.4 angstrom. Simple mathematical calculation. Now width or diameter of a double helix is 20 angstrom or 2 nanometer. So these are overall characteristics of B form of the DNA, that means Watson-Crick model of double helix DNA structure. Let's discuss about Wien-Chargaff's rule, which is only applicable for double-stranded DNA. It is not applicable for RNA. Already to some extent we have discussed about complementary base pairing in double-stranded DNA. In a double-stranded DNA, hydrogen bonds are formed between a purine, that means A and G, and a pyrimidine, that means T or C only. That means purine-purine or pyrimidine-pyrimidine hydrogen bond formation is not possible due to the unfavorable spatial arrangement. Only favorable spatial arrangement is possible in between adenine and T and guanine and cytosine. So in this figure you can see this is one hydrogen bond, this is second hydrogen bond. So total two hydrogen bonds present in between T and A, that means A and T, AT double hydrogen bond. But in between G and C, that means guanine pairs with cytosine through three hydrogen bonds. Here you can it is clearly visible total three hydrogen bonds can be produced in between G and C. But there is no possibility of purine-purine or pyrimidine-pyrimidine hydrogen bond formation, which is not favorable orientation or spatial arrangement. This complementary base pairing in DNA proves Chargaff's rule. It proves Chargaff's rule. Now, in case of, according to the Chargaff's rule, base pair equality, if you consider here, the amount of adenine is equal to the amount of thymine. So, in a double-stranded DNA, amount of A is equal to amount of T. Similarly, the amount of G is equal to amount of C. Because of the fact that A is only can bind with T and G is only bind with C. So in order to get the proper double helix structure, they should present in the equal amount. A should present in equal amount with T and G should present in equal amount with C. And from these two facts, we can also derivatize other facts like purine, amount of purine in a double helix structure of DNA is equal to amount of pyrimidine. And in that case, again, we can also write in this way: purine divided by pyrimidine is equal to 1. And as I told you, A is equal to T and G, A is equal to T and G is equal to C. So if you do addition of A and G and T and C, you will get A plus G is equal to T plus C. So we can get A plus G is equal to T plus C. And that means A plus G divided by T plus C is equal to 1. However, A plus T divided by G plus C is not equal to 1. It is actually constant. That is for a particular species. So species to species, A plus T divided by G plus C is constant. So the amount of A plus T need not always be equal to the amount of G plus C. And the ratio of A plus T divided by G plus C remains constant for a particular species but could vary from one species to another. So this is all about Arvind-Chargaff's rule. Nucleotides in DNA are linked together by two main types of bonds. First one is phosphodiester bond, that means 3' 5' phosphodiester bond, and second bond is hydrogen bond. Now, if you consider about the phosphodiester bond, these covalent bonds form the backbone of the DNA molecule, and they connect the 3' carbon atom of one sugar molecule, that is deoxyribose, to the 5' carbon atom of the next sugar molecule. Already I have shown that in my previous slides. Now, another one point is that the phosphate group forms a bridge between two sugar molecules, creating strong and stable chain. And this 3' 5' phosphodiester bond present in a single strand of the DNA. So in between two nucleotides, side by side two nucleotides, bond present is 3' 5' phosphodiester bond. But if you consider about double-stranded DNA, so in between two strands, connection between two nucleotides, that is two hydrogen bond, and these are weak bonds that occur between the nitrogenous bases on opposite strands of the DNA double helix. Adenine pairs with thymine through two hydrogen bonds, already you know, and guanine bound with triple hydrogen bond with cytosine. These bonds are critical for the stability and specificity of the DNA double helix, allowing for the complementary base pairing essential for the replication and transcription process. Let's discuss about differences between DNA and RNA. Full form of DNA is deoxyribonucleic acid. Full form of RNA is ribonucleic acid. DNA is double-stranded helical structure present in DNA, whereas RNA is mostly single-stranded. Nitrogen bases present in DNA, those are A, T, G, C, that means adenine, thymine, guanine, and cytosine. Whereas in case of RNA, A, U, G, C, that means adenine, uracil, guanine, and cytosine. So, difference only in thymine and uracil. Thymine present in DNA, whereas uracil present in RNA. If you consider about the sugar composition, so pentose sugar present both the cases, but in DNA it is deoxyribose sugar, and in RNA it is ribose sugar. Stability, if you consider, so DNA is more stable due to the double-stranded structure, and another reason is that it has deoxyribose sugar. Due to these two major reasons, DNA is more stable, whereas RNA is the less stable, mainly due to single-stranded structure, as well as it has a ribose sugar, which is more reactive as compared to deoxyribose present in DNA. If you consider about the function of DNA, it stores genetic information in all living organisms and in case of some virus. And if you consider RNA mainly it converts the information stored in DNA into proteins, and it also serves as the genetic material in few virus. Location of DNA, that is mostly found in the cell nucleus. If you consider about eukaryotic system, if you consider about prokaryotic system, it is present in the nucleoid, and some cases it is present in the plasmid of bacteria. In case of eukaryotic system, another place of DNA is in the mitochondria and chloroplast. They have different DNAs, and RNA is found in the cell nucleus, in the cytoplasm, or in the ribosome. Hydrogen bond occurs between the nitrogenous bases on opposite strands of the DNA double helix, and in that case A creates a double hydrogen bond with T, and G creates triple hydrogen bond with C. Whereas hydrogen bond is absent in case of RNA. Arvin-Chargap's rule is applicable only for DNA. In that case, a number of A is equal to number of T, and number of G is equal to number of C. Whereas as double-stranded structure is not there in RNA, so it is not applicable. Chargap's rule is not applicable for RNA. That's all about nitrogen bases, nucleic acids, nucleotides, and nucleic acids. That means DNA and RNA. Thank you very much for watching this video. In order to get a next video on biomolecules, don't forget to like, share, and subscribe this channel. Take care. Bye bye.